Sara's not here today to give her talk, so that's a bit unfortunate, but maybe it's better because her slides wouldn't have been here. The whole point of kind of having us in this room giving this talk is to try to bring a new perspective to thinking about exoplanet biosignatures. And it's actually a discussion that we really would be having these kind of methods in a different room at AbSciCon. So can we actually bring some of the techniques that we're using in different areas into thinking about exoplanet biosignatures? And so I am an original life scientist. I work primarily on thinking about living systems. And one of the things I think is really intriguing to me about exoplanets is the opportunity actually to think about life from an entirely different perspective. And so what I would hope is that we can actually take advantage of a unique opportunity that we're going to get with exoplanets to get more information about building theories for living systems. And so that's actually my ultimate goal. But what we're trying to do is actually look at atmospheres more from a systems perspective. And so what people do in other areas of science that are thinking about living systems from more quantitative perspectives is actually try to think about the network structure of living systems. And what I mean by this, so how many people here are actually on Facebook? A lot of people. How many people are tweeting right now? Okay. You're a part of a network. You may not know it, but you are. And so what happens on Facebook, if you want to represent that mathematically, what the structure of Facebook is, you can do that in a graphical representation. And so each person in this room that raises their hand, raise your hand again, you would be a node. How many people are in here are friends with other people in this room? Probably everybody. Yeah, okay. So there would be connections between the people in this room. And we could actually build a network structure to describe that. As it turns out, when you do that for living systems, when you do that for social systems, or you do it for chemical systems, you end up realizing that there's a lot of statistical regularities in that structure. And so that's actually the topological properties of the networks that we study. And so what I'm going to talk about at 2:45 in another room, which hopefully will have my slides, is actually the biochemical network structure of the biosphere as a whole. So one of the things that we've been doing in my group is actually looking at the structure of reactions within individual organisms at the level of entire communities and at the level of the biosphere as a whole. And they have a lot of statistical regularities across those scales. As it turns out, some of those statistical regularities are also apparent in atmospheres of planets. So some network theorists, being network theorists, they like to study systems in all kinds of different areas, have actually published a few papers looking at the network structure of chemical reaction networks in atmospheres. And I think this is a literature that people in the exoplanet community maybe are not aware of. It's a very small literature. I've only been able to come across less than 10 papers doing this. But I think it's an incredibly promising avenue for thinking about atmospheres, not just in terms of their molecular constituents, but their actual structure. Like, what is the structure of an atmosphere from this kind of perspective that's relevant to living systems? And so I think I'm just going to go without my slides at this point. I think I have to. Okay, so I'm just going to, you guys are just going to get a teaser of, this kind of new approach to thinking about biosignatures. And then really, actually, the whole point of this was to open this as a conversation for thinking about things a little bit differently. So these few papers have actually demonstrated that there might be some features, just looking at the atmospheres in our own solar system, that are different between Earth and other atmospheres. And so for those of you that raised your hands for Facebook, raise your hands again. Okay, so what kind of statistical regularities am I talking about? How many people in this room have more than 100 friends on Facebook? Okay, how many people have more than 200 friends? Okay, how many people have more than 300 friends? How many people have more than 400 friends? I'm not raising my hand anymore. I was lost long ago. I'm one of those, wallflower. 500 friends? Okay, so what we just saw was actually a distribution of how many friends people have. And you can look at those kind of distributions, and if you look at them in biochemical networks, they actually have a power law distribution. And that kind of property is one of the kind of properties that we think is actually relevant to biological organization. And so if you look at Earth's atmosphere, if you actually inventoried how many reactions molecules participate in Earth's atmosphere, it would follow a similar distribution to how you guys raised your hands. But if you look at the atmosphere of Mars or Venus or Titan, the preliminary analysis people have done suggests that it's not really quite that distribution. It might be more homogeneous. So there might be more people with 100 friends. Everybody has the same number of friends. And there aren't those anomalies that have 1,000 friends. So the heterogeneity of the distribution of having really popular people, having Justin Biebers, for example, is really indicative of living systems. It leads to a robustness in living systems, and it actually could potentially be a biosignature. And so what we're interested in doing is actually doing more rigorous analysis of different planetary atmospheres to try to figure out if this really is a robust biosignature, if it's not just an artifact of our models. What are the conditions that give rise to this kind of structure in Earth's atmosphere that makes it different from the atmospheres of other worlds? And how can we actually use that in exoplanets? Can we directly detect this kind of structure at a systems level? And so it becomes a really kind of interesting problem for understanding atmospheres and how they're coupled to a biosphere, because the way I actually think about it is you have this network of all the chemistry on the surface of a planet that biology catalyzes, and you have an atmosphere, and those are actually two coupled networks that are overlaid over each other, and one is driving the other, right? So can we actually think about them at that global scale of the reaction network structure? And so that would actually be the ultimate hope. So I'm going to finish my song and dance routine because I think I could go on for this forever. But actually, since we have the time, maybe it'd be good to have like some community comment, questions, discussion. And yeah, we'll see how that goes. How much more time do I have?
You including questions, have about 9 more minutes.
Wow.
But first, let's give you a really deep round of applause. That's really impressive without slides. And everyone will remember.
So I'd actually really like just some, yeah, thanks, Bob. Come on up.
Questions, please.
Yeah.
So this is so cool, and it's so ironic because what you're talking about is the ultimate kind of visualization of data, and you can't see it. So we've been doing a similar thing with minerals, and we find that we think we're also seeing a biosignature on Earth with the distribution of minerals. In our particular case, it's a particular frequency distribution called the large number of rare event distribution, but it seems like that's not found in any other, at least terrestrial body in our system. We also are seeing really interesting network topologies, which is a different thing. And again, I mean, that's the power of networks. You're seeing lots of different dimensions. So do you have any of the network metrics that you can tell us about?
Sure. Yeah, so I'm really glad that you're looking at minerals. And so one thing I'd like to do is start connecting like the Earth's geochemical networks to biochemical networks to atmospheric networks to do this kind of analysis. But the kind of things we're looking at, so traditionally people look at the degree distribution. It's a technical term for that hand waving that we just did, which is actually looking at how connected individual molecules are and then plotting that connectivity in a rank order fashion and fitting it with a nice power law. But it turns out to be really hard to fit a power law to networks in a statistically rigorous way. So I am more in favor of using other measures besides just the degree sequence for trying to understand the structure of these networks. So some of the things that we've been looking at are things like the clustering in the network. So for example, if I'm friends with Sean, and Sean knows Hillary, and Hillary knows Nancy, and Nancy knows Sean, they form a triangle among my friends, and I can count how many triangles among my friends there are in the system, and that actually becomes a statistical property of the network. And so we can look at that. We've been looking at average path length, so you can look at how far it takes to get from one individual to another. So how many chemical reactions does it take to get from one species to another species in the network? So probably most people in this room have heard of like 6 degrees of separation. That's actually where that comes from, is from network science, that you actually literally in a social network are only 6 people away from anybody on the planet. And that was an experiment that was done that was pretty cool. So that's another one. And so there's all these kinds of, we could get into more technical conversation later, Bob, because I'd really like to hear more about what you're doing, and I missed your talk on that this morning. So that'd be great. I'll go to the next question, though.
Hi, Joshua Chrysanthemum, University of Washington. So I might be wrong about this, but I suspect that a lot of the complexity in the network of Earth's atmosphere is attributable to trace biogenic species. And so I'm wondering about the detectability of these sort of network properties for Earth-like exoplanets. What would you look for?
So interesting. This is a great question. So I think even if you had trace species in the network, they could change the structure of the network enough to be detectable. But a lot of the stuff that we're looking at is global properties of the network. So they will be things that are statistically averaged over the entire network. So you'll count, for example, how many friends each individual in the network has, and then look at the average over the entire network. You can also look at those as local measures and how they're distributed in the network. But since you're trying to infer the properties of the structure of the network as a whole, it's not as sensitive as you would think to individual species. And so it is in principle possible, maybe because we're looking at it in a statistical sense, to be able to infer those properties. Does that make sense in the context of your question? Okay.
Hi, Emma Jablonski.
Hi.
I have a somewhat related question. You said that there is a difference between the network artifacts in Earth versus some of the other planets or other. Is any of that attributable to the amount of data available?
Yeah, so actually that's a big problem. And that's one of the things that we're actually actively working to try to get to the bottom of. And so since I didn't have my slides, I didn't really mention my collaborators on this. But besides Tessa Fisher, there's 2 other students in my group, Harrison Smith and Coral Ruiz, working on this. And we also have Mike Line and Jim Lyons at ASU that are giving sort of atmospheric expertise, which is one of the nice things about ASU. We have a network scientist next to an atmospheric scientist in the same hallway, which is kind of fun. So the reason I mention that now is that Jim and Mike are trying to help bring sort of the expertise about the biases in different models, because there are a lot of them. And one of the bigger problems is that the network for Earth is huge by comparison, say, to what we know of Mars, for example. And so that's actually one of the reasons I don't trust the degree sequence, for example. So if you want to fit like a power law to a network that has 30 nodes, like what we look at with Mars's network that we've been analyzing so far, compared to one that has a couple hundred nodes like the Earth network, your fits are much more reliable for Earth than for Mars. So I think what we need to do is look at the properties that are not necessarily as dependent on how large your network is. So that's one. Another one is that a lot of times with these, so even when you're looking at biochemical networks, you can construct those from genomic data, but oftentimes we don't have all the genomic data. And there are a lot of analyses done on these kind of networks to show that they're robust in terms of these sort of like global measurements that we're taking to say if you knocked out 10% of nodes, to try to represent the fact that maybe we don't know 10% of the genes in a genome, and so how accurately are we actually getting a picture of that structure of that network. So they are robust to sort of a certain degree of missing data, but it depends on how unknown your unknown is. So if we're missing several hundred reactions and we only have 300, then we have a big problem. But if it's, or 30, but if it's 30 and we're missing 10, then it's not so problematic. So yeah, so that's where I think this is fruitful to start having these kind of conversations, because from the network side, we know how to do these analyses and relate them to what's the story in biology, but from the atmospheric side, I really rely on my colleagues to tell me what models are actually giving an accurate picture of an atmosphere and what's missing.
You have time for one more question.
A fantastic talk, great graphics.
Thank you. It was on your mind.
I know.
Those are the best graphics of all.
So as I understand, the sort of power law distribution for the network of Earth means you've got one or two species that react with a huge number of different things.
Yeah, so you have a few species that react with a huge number of things.
Okay, so isn't that just oxygen?
Yes.
So what you detected is that Earth has oxygen.
Yes. So oxygen is a huge bias in the structure of the network. And so one of the questions that we have is maybe the structure of modern Earth's atmosphere completely attributable to oxygen, or does it involve other biogenic gases, and how is that actually driving the structure? And we have looked at some models for early Earth and don't have the data to show, but early Earth networks tend to be heterogeneous too. They're just not as heterogeneous as modern Earth. So I think that there still are some opportunities besides the oxygen story to look at that. And so one of the things that we'd like to do moving forward is look at different classes of chemical species and see how they change the structure of networks to try to build up more of a theory that is coupled to the way we think about chemistry and atmospheres and the way we think about the network structure that those chemistries. So you could actually have more of like be able to predict based on certain sets of molecules being present what that kind of structure should
Well,
be.
if you want a big list of gases,
I
we've got one
know.
for you.
That's why we're going to talk.
All right, another resounding round of applause for Sarah, please.
Thank you.